So hello everyone, good afternoon and welcome to today's webinar. My name is Ben and I'll be talking you through the Ensembl Rapid Release webinar. So the focus of today's webinar is the Ensembl Rapid Release Genome browser. This is actually something that's relatively new within the Ensembl Genome browser site. So we have the main Ensembl Genome browser, which you may have been using in your work already. But the Rapid Release Genome browser is slightly different. There are different species and different data available. So I want to take some time today to go through those species and what that data is and show you how to use that browser using an example. So what I want to cover today is what the Ensembl Rapid Release Genome browser is, the different types of data that we can find through the Rapid Release Genome browser, and how to navigate the genome browser itself. There is many similarities to the Ensembl Genome browser itself, the main Ensembl Genome browser, but there are some slight differences. So I'm going to try and pick up on those and show you how to navigate and get the data that you need. So just to follow on from Anna's introduction, I'll be talking you through the Rapid Release Genome browser in today's webinar. If you do have any questions, then you can ask those in the chat box in the GoToTraining interface. So my colleagues Astrid and Michal will both be there in the chat box waiting for you to ask any questions. There's no threading within the chat box, so obviously if there's lots of questions all at once, that can make it quite tricky to sort of track all of the questions and the individual responses. So what you should look out for is either Astrid or Michal replying with an @ within your username. So they'll be able to answer you directly, but they'll post their answers to everyone for everyone to see. At the very end of the webinar, we'll circle back round and see if there are any final questions that I can quickly go through before we finish up. So I want to start by introducing you to a brief history of the Ensembl project and how we have reached the point of needing a rapid release genome browser. So the Ensembl project was first conceived of as an offshoot of the Human Genome Project. So in January 2000, around the same time as the release of the first draft of the human genome sequence, the Ensembl project and genome browser was launched. And at that time, all that the Ensembl genome browser contained was human data, so the human genome and annotation around the human genome assembly. By January 2005, we'd increased in size, including data from important model organisms. So as well as human, there was also the genome assemblies and annotation for mouse, zebrafish, fruit flies, C. elegans, the nematode worm, and yeast as well, Saccharomyces cerevisiae, as well as 8 other important vertebrate species. In January 2008, a separate sister project was launched called Ensembl Genomes. So again, related to Ensembl, but in fact, this is a slightly different project in that the data that was being presented in Ensembl Genomes was for non-vertebrate species. So bacteria, protists, plants, fungi, and non-vertebrate metazoans. So Ensembl and Ensembl Genomes were existing at the same time together, one presenting data for the vertebrate species, that's Ensembl, and then Ensembl Genomes presenting data for the non-vertebrates. By October of 2016, so around about 4 years ago, the Ensembl genome browser, so looking at vertebrate species, passed over 100 different vertebrate species contained in our databases. And then earlier this year, in April 2020, Ensembl Release 100 had 286 vertebrate species, so nearly 300 species. And so basically you can see over these 20 years how the number of these species in Ensembl in particular has been growing quite rapidly. And that's obviously a technical problem that we have to solve in terms of being able to store and present such a wide array of data for such a large number of species. So for each of these species, we have a lot of data associated with them as well. So for each of the species in both Ensembl and for all the species in both Ensembl and Ensembl genomes, we present the publicly available genomic assemblies with gene annotation that's either generated by ourselves using our own automated gene annotation pipeline, or for the non-vertebrates imported from the different communities that have produced the genome assemblies and annotations. We also present variation data. So within Ensembl, there are around about 20, 21, 22, 23 species that have variation data available. So we import millions of variants for a number of different species, which include both small and large scale sequence variants with phenotype associations as well. And we also import variation data where it's available for non-vertebrate species in Ensembl genomes as well. One of the other data types that we generate is called the comparative genomics. So this is where we compare the genome assemblies and the gene models from one species to another. So we perform analyses such as whole genome alignments, and we predict orthologous relationships by creating gene trees, for example. And we do that for all of the species in Ensembl and Ensembl genomes, and we even have a pan comparer where we also perform some comparative genomics analyses between the vertebrates and non-vertebrate species as well. And finally, we also have a regulatory build. So this is where we predict promoters and enhancers, so those features that regulate gene expression for human and mouse. So this is actually only a data type that's available for 2 species at the moment, although in the future this is going to be a data type that's also being produced for a number of agricultural species as well, for example, and model organisms. So the point that I'm trying to make is that as well as the number of species that we are presenting in Ensembl and Ensembl genomes, which is increasing over time, for each species there's also a large amount of data that we're trying to store and present as well. So in order to keep up to date with the latest data that's being produced by the scientific communities and consortium around the world, Ensembl releases a new version every approximately 3 months. So at the moment, the current Ensembl version is Ensembl 101. That was released in October of 2020, in August of 2020. And obviously, at that time, we had a certain set of data in our databases, which was frozen in time and released in August. At the moment, we're currently looking for new genome assemblies for different species. We're updating our underlying software. We're updating any gene annotation according to new evidence, new data that's being produced. We reperform our comparative analyses based on the new species that are being loaded into our databases. We update and import new variation data where that's available. We update our regulatory build, our regulation data, and we also update our web interfaces as well. So what we do is we package all of that new data up and we release it as a new version. For example, Ensembl 102 is going to be released in October of 2020. So hopefully in just a few weeks' time, Ensembl 102 will be released. And what this means is that all of the data in Ensembl is very traceable. So you can look at a particular version of the Ensembl databases and know what data was there at what time. So we make data available through our archive, so you can go back in time and check and see what data there is and what data there was at a specific point in time. But this release cycle allows us to incorporate new data, new species, new genome assemblies in a relatively short time frame. So every 3 months we have new data being added to Ensembl. However, the problem is that this 3-month release cycle is actually finding it difficult to keep up with the amount of data that's being produced around the world all the time, essentially by large collaborative projects that are aiming to sequence many species all around the world. So just as an example, the Darwin Tree of Life is a project aiming to sequence 66,000 eukaryotes across the British Isles. Then we have the VGP, the Vertebrate Genomes Project. That's aiming to sequence every single vertebrate species, to sequence the genome of every single vertebrate species on the planet. So that's around about 70,000 species that are going to have their genomes sequenced and then hopefully loaded into the Ensembl databases. And then there's the Earth Biogenome Project, which is aiming to sequence 1.5 million eukaryote species around the world. So obviously the different species perhaps in these projects are slightly overlapping, but the idea that I want to impress upon you is that in perhaps just a few years' time, instead of Ensembl containing 200, 300 species, we're going to be containing thousands of species. And this data is being produced at such a rapid rate that it's important for us to it's important for us to be able to have databases that can handle this amount of data, but also to release it in a timely manner so that the data can be accessed by users around the world as quickly as possible. So that's why we've launched the new Rapid Release Genome browser. So the idea with the Rapid Release Genome browser is that it's a lightweight genome browser that contains a small amount of very important data for newly sequenced species. So you can see the web address of the Rapid Release Genome browser at the top there, rapid.ensembl.org, and that's the website that we're going to visit in just a moment. And what we can see on the left-hand side of the screen here is this is a screenshot of the homepage, which we're going to explore in a moment. And just on the right-hand side, I've listed the main data types that we make available through the Rapid Release Genome browser. So for each species, for each genome assembly that we present, there's a gene annotation with Ensembl stable IDs. So the genes themselves are annotated using an Ensembl automated pipeline and then assigned stable IDs. We also have repeat annotation. So repeats within the genome are annotated and we can view those in the genome browser. The protein features across transcripts and the proteins that they encode are annotated as well. We have BLAST functionality, so we have a BLAST tool available for you to perform some sequence-based searching. And finally, we also make file dumps available through our FTP site. So this includes transcripts and protein sequences in FASTA format, the soft-masked genome sequence, as well as other file types as well available through the Rapid Release FTP site, which we're also going to explore today. So what I'd like to do in the rest of this webinar is to look at an example gene. We're going to use a brown bear gene here that you can see. This is the Ensembl stable ID, ENSUARG, and then a unique 11-digit number. That's the Ensembl stable ID. That is for the brown bear gene of interest. And then we're going to manipulate the views to see the data that we're interested in. So I'm just going to jump out of my presentation and I'm going to go to a new tab in my internet browser where I'm going to type the web address for the Ensembl Rapid Release website. So it's rapid.ensembl.org. And that takes us to the Ensembl Rapid Release website. So if you're familiar with the Ensembl Genome browser, you'll see that in fact the layout is probably very similar to what you are used to exploring already, so what you've seen before. So what you can see at the top is a navigation bar. This blue bar at the top will appear at the top of every single page within the Ensembl Rapid Release Genome browser. And you can see the links there that help you navigate around the website. So the logo is actually a link to return back to the homepage. Then we have links to to the BLAST tool. So if you want to perform some sequence-based searching against any of the genomes in Ensembl Rapid Release, you can click on BLAST. There's also information about downloading data, which we're going to cover when we look at the FTP site as well. And then there's also some help and docs as well that you can explore as well in your own time. Over on the right-hand side, you can see that we have information about the latest genomes that have been included. So the latest genomes, there's 8 species that are included in the latest release. And what you'll find is that this list is updated approximately every 2 weeks. So the Ensembl Rapid Release genome browser is released approximately every 2 weeks, compared to the main Ensembl site, which is released every 3 months. So this, you can really see how quickly the data is going to be produced and then made available, so every 2 weeks. Over on the left-hand side of the page, you can see we have a search bar. So this is going to allow us to search for genomic regions or genes within our species of interest. And then further down the page, you can see we have information about the genomes themselves. So you can select individual genomes and find information about them. So that's what I want to do now. I'm going to click on the link here, view and download available data for all species. So you can use the drop-down list here, but obviously this list is going to be very long. We're planning to include a lot of species in the Ensembl Rapid Release genome browser. So it's actually normally easier to view the table and then begin filtering down and searching. So if you click on the link here, view and download available data for all species. This will take you to a page where you can find a table with all the species. Large, searching through it, and you can use the filter in the top right-hand corner here to search for your species of interest. So you can see in the table we have the scientific name, but also the common name of the different species. If this column is not showing for you, you can show and hide the different columns here. So if the common name is not showing for you by default, you can click here and find the common name. You can also turn on the other tracks, such as the taxon ID, and get the assembly name and the genome size as well, for example. And you can also find the gene count as well. You can add that if you want to find the coding gene count as well. So as you can see, this is now my table. I can perhaps begin filtering by searching for a species of interest. So maybe I'm interested in the brown bear. So I can just begin typing brown, and you can see already that my table is being filtered. I've got the brown-headed cowbird. Obviously, it's just found the word brown in my search term, but importantly, I have brown bear underneath here. So this is my species of interest, brown bear. It's a mammal. We've got the assembly name here as well, and the accession. So this is the unique accession number from the ENA archive, where this genome assembly was deposited. We've got the genome size. You can see that the Ensembl project was responsible for performing the annotation of the genes. And then we've got a count for the number of coding genes across the whole genome. The columns on the right-hand side of the table show us, provide us links to the FTP site to actually begin downloading data files, genome-wide data files for these different species. So you might not be familiar with all of these different data types. You can see here, for example, we have annotation data available in GTF and GFF3 format. We also have protein sequences available in FASTA format, as well as the soft masked genome sequence available in FASTA format. So if you want to export these files, for example, you can just click on them. I'm going to right click and open in a new tab. So you can see here that takes me to the FTP site. You can see the link at the address at the top here, ftp.ensembl.org/pub/rapid release. So this is the directory where the Ensembl Rapid Release files are stored, and you can download them. So if you want to download this GTF file, you can obviously just download it and use it in your analyses. As I've mentioned, we also have the protein and the genome sequence in FASTA format. And there are some other files available, different formats for different data types. So you can actually click here on FTP under the other files column. And you can see here, this just takes us to the main directory, the rapid release directory. So you can see these are the different file types that we make available. So if you want to download any FASTA files, for example, you can click FASTA. Then you'll need to find your species of INAIC name. Maybe we can use this as an example. And then you can see we have the cDNA, CDS, DNA, non-coding RNA peptide sequence as well. So you can, for example, download the FASTA file of the cDNA sequences of the species. Okay, very good. I'm just going to use the back button in my browser to go back to our species table. And I'm going to search for brown bear again. Okay, so if we want to look at a gene of interest, for example, for the brown bear, we can click on the species name. That will take us to the species-specific homepage, where we have information about the genome assembly. For example, over here, you can see the links with the ENA accession number and the length of the genome, for example, and the gene counts. Then we have a search bar. So we can actually put our stable ID of interest into the search bar here. So this is our ID from our example, and then we can click go. And then this takes us to a results page. So this is just a summary of the different pages that have information about this feature that we searched. So we can click on the gene ID. And this takes us to the gene tab. So just as we have in Ensembl, the main Ensembl site, all of the data is organized into this tab system. So at the moment, we've searched for a gene. So we're looking at the gene tab. When we look at the individual transcripts of this gene, we'll go to the transcript tab. And then eventually, we're going to have a look at the genomic location itself. So we're going to have a look at the location tab. So in the gene tab, you can see that we land on what we call a summary page, where we have information about the location of this gene, the number of transcripts annotated for this gene, and we can choose to either hide or show our transcript table. So we can see these are the different transcript IDs with their length and their biotype. So you can see that this gene has 4 protein coding transcripts. And you can see underneath, we can see these transcripts represented as these boxes and lines. So each of these each of these boxes and line diagrams represents a single transcript, where the boxes are the exons that I'm just hovering over here. You can see these are the exons, and then the lines are the introns. You can see that the transcripts of this gene, they're all on the reverse strand. You can see the arrow here indicating the direction of transcription. So this is the 5' end of our transcript of interest, and then this is the direction of transcription with the 3' end over here. The next thing to notice is the menu over on the left-hand side. So you can see that there are a number of different data types that we can explore for this gene of interest. So we can firstly look at the sequence. So we can see, if we scroll down, we can see this sequence of this gene in FASTA format. So this is actually the genomic region where this gene exists. And we can see the exons highlighted in this peach coloring, the peach highlighting, with the exons of this gene with the red lettering. So you can see here, this is the first exon of our gene of interest. And then this is intronic sequence. The other exons of this gene. You can download this sequence if you want to, so you can just click download, and that will open up a pop-up window which will allow you to export a number of different types of sequence. So you can choose to download the genomic sequence, or you can choose to download the cDNA or the coding sequence, depending on your own interest and your own research questions. So you can download those sequence those sequences in FASTA format. That's the format that's selected by default. You can see that at the top here in the drop-down menu. File format is FASTA by default. But you can also choose the RTF format. So this is the rich text format that allows you to export the sequence with it's more of a static image. So it's not so useful for perhaps copying and pasting into primary design tools or any other pieces of software that you might be using. But it does present to you the annotation on the sequence. So it will present the highlighting of the exons within the sequence. And that might be useful for you to use as a reference, perhaps when you're looking at the sequence and trying to work out where the exons are within the sequence that you just downloaded. You obviously would then need to click download, which I won't do now, but you would do that if you want to download the sequence. When you're happy, you can close the pop-up window by clicking on the tick in the top right-hand corner. And then we can begin just to explore some of the other data that's available. So the species in the Ensembl Rapid Release site, we also have gene ontology annotation where that's available. So the gene ontologies try to explain the function of the gene using standardized vocabulary. So what we can find here, if we click on Go Biological Process, are the biological processes that this gene and its transcripts are annotated to be involved in. So you can see that this gene has been linked to peptidyl arginine methylation, and this is the evidence that's been used by the Gene Ontology Consortium. So it's been inferred by electronic annotation. We also have molecular function. So we can see here that the transcripts of this gene have been involved, well, are predicted, annotated to be involved in protein binding, methyl transferase activity, and protein arginine and methyl transferase activity as well. So that's the information that we can really find through the Gene tab. We can also find information through the Transcripts tab. So what we need to do to find the Transcript tab is to pick one of the transcripts from the transcript table at the top here. We'll just choose this first one as an example, the one that's 1,305 base pairs in length. This takes us to the Transcript tab. So you can see here a new tab is opened at the top here. So all the information on this page is relating to this specific transcript of this gene that we're interested in. I'm going to hide the transcript table now just to make the view a bit more clear. And we can again see this graphical representation of this transcript with the exons and then the introns depicted as these boxes and lines. Again, we have this information. We have further information through the menu over on the left-hand side. So we can view the sequence in different formats. So we can view the sequence of the individual exons of this transcript. So we can see here the individual exons within this table. So we have exon 1, for example. We have its start and end position, the phase, the length, and then the actual sequence itself. Then we have some intronic sequence available here. Obviously, we don't show the full intronic sequence. We just show you the sequence at the exon intron boundary. But if you click on configure this page, the blue button over on the left hand side underneath the menu, you can choose to edit the sequences that you view on this page. So you can choose to view more flanking sequences, or you can choose to show the full intronic sequence, for example. So there's some different options that you can select here, and when you're happy you can save and close. And now you can see, in fact, we also have the full intronic sequence case. And again, we have now exon 2 and then another intron further down. Again, we can download this sequence just as before. And then we also have over on the left-hand side in the menu, the cDNA sequence. So you can see the cDNA sequence is represented here. We have the genomic sequence in row 1 with the exons in black here, in the blue color, in the black and blue alternating colors. Sorry, so we have exon 1 in this black color here, and then exon 2 in the blue lettering here. Then we have the cDNA sequence underneath. So we have the ATG. You can see the start code on here, and then the final row for each sequence is the peptide sequence. So this M is the methionine that's encoded by this ATG code on here. And again, just as before, you can also download the sequence if you want to download the cDNA sequence. And finally, we also have the protein sequence as well, which you can view and download as before. The final piece of information that we can get through the for the transcript is the protein features. So we can click on protein summary from the menu on the left-hand side. And we can see the different domains and motifs that are predicted to exist within the protein that's encoded by this transcript. So you can see, for example, there's an SH3 domain that's predicted by superfamily, SMART, PFAM, and ProSite profiles, all towards the N terminus of our protein. So this pink and purple bar represents the protein itself. So we've got the N terminus of our peptide over here on the left-hand side, and the C terminus of our peptide over here. And then you can also see that there are other domains predicted, for example, a methyl transferase small domain predicted by PFAM in this region of the protein as well. If you click on domains and features in the menu on the left, you'll find the data that we saw on the previous page, but just in a tabular form. So you'll be able to find the accession IDs of each of these features, and you can obviously use these links to go out to the various resources such as Interpro, for example. So the final thing that I wanted to show you for each of the for our gene of interest is that we can view this in the location tab as well. So when we opened our gene tab, one of the other tabs that opened by default was the location tab. So if we click here from location on location near the top in the blue bar, we can see that this opens what we call the region in detail view. So this is the genome browser, and it's been automatically loaded to show the region of the genome where our gene of interest exists. So what we can see here is that our page is split into 3 separate images. So at the top we have a chromosomal overview. So depending on the species of interest, the genomes might not be assembled into individual chromosomes. They might still be assembled at a scaffold level. So this, for example, might just be a single scaffold or a chromosome, depending on the species. And then our red box indicates our region of interest. Then as we scroll down, we begin to see the genome in more detail. So in the image underneath, we have this 1 megabase overview. So you can see the scale at the top here. And you can see the data is now beginning to be arranged into what we call tracks. So we can see the contigs track here. This is the blue bar. So the name of the track's on the left-hand side here, and the data is represented as this blue bar. So the contigs track represents the genome assembly itself. So this blue bar represents double-stranded DNA, with the forward strand moving left to right in this direction, and then the reverse strand moving right to left in a 5' direction. Then underneath we have the genes track. So this is a separate track that's showing us all of the genes that are annotated in this region. And then we have our red box, which is indicating again our region of interest. So we're just beginning to zoom in on our red box, our region of interest, and you can see, in fact, this is our gene of interest that we originally searched for, this red block with the ID underneath. And then there are genes that are further downstream, for example. At the very bottom, you can see that we have the region in detail itself. So again, we have data arranged into tracks. So we have our contigs track here, representing the double-stranded DNA. And then we can see the genes track. This time, the genes are not represented as the blocks, as we saw above, but they're represented as the individual transcripts for each gene. So again, this gene has got 4 transcripts, and you can see each of these transcripts has a different exon and intron patterns. So what we can do when we're looking at this region in detail view is we can actually add tracks to view, and we can add tracks to this image, and that will be we can view it alongside this other data that we already have loaded by default. So if we go back up the page and click on configure this page, this will show us all of the tracks that we can add to this view. So what you can see is that by default, we're looking at active tracks. So these are the tracks that are shown when we first arrive on the page without any modifications. But you can also see that there are other tracks available. So we have sequence and assembly. The contigs and sequence tracks are shown by default. You can see that because the boxes have got the icons there already. But these tracks here, the translated sequence and the start-stop codons, these are not shown by default. So if you want to turn them on, you can click on the box to turn on the different tracks. If you want to add repeats, so for example, we know that we are able to view repeats that are annotated across the genome. There's a section in the menu on the left-hand side called Repeat Regions. So again, we can see here, these are the different repeats that we can view. So I'm going to click All Repeats. I'll click on the box to turn the track on, and then click Expanded as the format. So now this track is turned on, so I'll be able to see all of the repeats that have been annotated in my region of interest. From this pop-up window, just to point out as well, it's also possible to upload custom data if you want to, using if you have your own data set that you want to view alongside the publicly available Ensembl data, you can do that with the custom tracks button there as well. So once you're happy with the tracks that you've added, you can save and close using the tick in the top right-hand corner. And now you can see that we have the tracks that we added, that we had there by default. So the contigs and the genes track showing us the transcripts. And now we also have the repeats as well, which we can hover over and click on to get more information. And the same with the transcripts as well. We can click on those and get more information from a pop-up window, including links back out to the protein or the transcript that we've already been looking at. Obviously, if you're perhaps particularly interested in maybe this exon here, you can use the options in the top right-hand corner, the toolbar here, to begin zooming in and scrolling around the genome. So if you have this dotted box selected, if you click and drag, you'll draw a box in your region of interest, which you can then jump to using the pop-up window. And this will help you zoom in to a region perhaps of particular interest that you are beginning to research, but beginning to investigate. And you can see now that we have the sequence up here as well. So this is the other track that I added. We can see this, the ATs, Gs, and Cs represented as the red, blue, green, and yellow bars. Good. Okay, so that brings me to the end of the demo of the Rapid Release Genome Browser. I'm just going to jump back to my presentation and wrap up, and then we're going to have a look to see if there are any questions that have not been answered so far from the presentation and demo. So just to summarize what we covered today, the Ensembl Rapid Release site is a lightweight genome browser that integrates some basic gene annotation data for recently sequenced species. And this has been mainly in response to the large amount of genome assemblies that are being produced by large collaborative projects and individual research groups around the world. And the data is all available through rapid.ensembl.org, that's the genome browser, or through ftp.ensembl.org/pub/rapid release. So that's the address for the FTP site, the Rapid Release FTP site. If you want further help and documentation for using both Ensembl Rapid Release Genome Browser, but also the Ensembl and Ensembl Genomes Genome Browser as well, then you can use the EBI online courses that you can find in the link here. We also have a number of YouTube tutorials and Youku. We upload our tutorials to Youku as well for those people who are unable to access YouTube. You can find lots of recorded webinars, presentations, tutorials for learning how to use different data types and different interfaces and tools within Ensembl. And if you do have any questions about using Ensembl, whether it's Ensembl Genomes, or the Rapid Release Genome Browser, for example, you can always email our help desk. So that's helpdesk@ensembl.org, and we'll always get back to you trying to help you understand the data that you're looking at. If you have questions, we'll work with you to try and resolve your queries. If you do use Ensembl or the Rapid Release Genome Browser, for example, in any of your work, then we greatly appreciate it when you cite us. So we have an annual paper that's released in NAR every year. So our latest one is called Ensembl 2020, and you'll be able to find the link, the DOI there for being able to cite Ensembl in your work. So we like it when people cite our paper because it helps us to know who is using Ensembl and what type of research they're doing. It helps us to develop our interfaces and our tools for the future as well. So obviously this is a very short webinar that's about one very small aspect of the Ensembl project, but we do have courses that are available for us to deliver at your institute that can cover any range of topics that you might be interested in, perhaps a general Ensembl genome browser workshop. Obviously at the moment we're not doing any face-to-face workshops, but we can still deliver virtual courses via Zoom or any other platforms about the Ensembl genome browser, the REST API as well. So if you have perhaps a desire to arrange an Ensembl workshop at your institute, you can again email the help desk. This webinar is obviously being held as part of the EBI webinar series. So there's actually more coming up later on this autumn in October and November and December. So if you want to find webinars about other topics, you can find those through the link there, ebi.ac.uk/training/webinars. And then, as Anna mentioned at the very beginning, we'd be very grateful if you filled out the survey that launches at the end of the webinar as well. That will help us give us feedback so that we know about the different topics that you would like to see being presented in webinars, and whether it was interesting and how the software worked for you technically. So this is the Ensembl team. We're around about 100 people, and obviously it's all of their hard work that goes into releasing all of the data through the Ensembl website and the Rapid Release Genome Browser as well. So the work I've been presenting today, the Genome Browser, the Rapid Release Genome Browser, is the hard work of all of these people. And you can also follow us through social media. So we have a Facebook page that you can follow. We have Twitter accounts that you can also follow, where we tweet about interesting data and ways that people are using Ensembl. And then we also have an Ensembl blog, www.ensembl.info, where we post information about job adverts, maintenance downtime, interesting announcing new releases, interesting new data that we're including. So if you're going to use Ensembl a lot, then I'd really suggest following the blog. It's quite a nice blog to follow to keep up to date with all of the data that's being produced by the Ensembl team. And that brings me to the end. So this is just our acknowledgement slide. So the list of people is basically essentially everyone that you'd have seen on the slide before. And then at the bottom, you can see these are our funders who obviously make the Ensembl project possible, and they ensure that the services that we provide are freely available for everyone to use around the world. Okay, very good. So I think that brings me to the end of the webinar. I'm just going to go across and open up the chat box. I can see that there have been a number of questions. I think some of them have been answered already by my colleagues Astrid and Michal. I'm not sure.
Yes.
AJ, I think that you were quite keen to read some of these out to me, perhaps?
Yes. Yeah, thanks a lot, Ben, for delivering the webinar today. And I would like to thank all the attendees for joining us today. Yeah, so as Ben said, some of the questions have been answered already, but we have one question. It's about how to retrieve 2 kb upstream DNA sequence for promoter analysis from transcription start site of the cDNA.
Okay, so that's quite a good question. There's actually a number of different ways that you can do that through the genome browser, the rapid release genome browser. So what I'm going to do is just go back to my, I'm just going to minimize my presentation. I'll go back to the genome browser. So we can do that perhaps from the transcript tab. So we can see here, for example, this is the transcript tab. We've got the sequence, the different sequence views over on the left-hand side. So we can click firstly on exons, perhaps. And we can see here that this is exon 1. So we can see here this is ATG. So this is actually the start codon that encodes the methionine. So there's no UTR sequence annotated for this transcript. So in fact, the first codon is the beginning, is the transcription start site. And we can see actually here, just before the first exon sequence, we have this green lettering, which is our flanking sequence. So this is actually the sequence that precedes our start codon. And we can increase the length of this flanking sequence if we click on configure this page. So we can see here, for example, flanking sequence at either end of transcript. So we could increase this to perhaps 2000. And now we have 2000 base pairs upstream of the start codon. And if you download this sequence, you could click download. And then you could get, for example, the genomic sequence, the exons, and then you can even here in this interface as well toggle the amount of flanking sequence that you see. There's a few other ways that you could do this. So again, with perhaps if you go to the gene tab, you can do the same thing in the sequence tab, in the sequence option in the menu on the left-hand side. Again, you can see that we have our first exon here with the ATG start codon. Again, if we click on configure this page, we can use the options in the pop-up window to toggle to change the amount of sequence that we see. So again, we could choose perhaps 2000 base pairs in a 5' flanking region, and then we again save and close. So we have this now 2000 base pairs preceding the start of the first exon. Again, as we did before, click download, and you can choose to download the genomic sequence with 2000 base pairs upstream, for example. Finally, you could also do this in the location tab. So when you're in the location tab, you can see, for example, perhaps we are looking, well, I zoomed in, so we're looking at a very particular, we're looking at a particular exon in one of the transcripts of the gene. But what we could do is we could click on our gene of interest. Or maybe if we navigate to the coordinates of our gene of interest. So I've just used the pop-up window just to go back to the view where I was viewing the full length of the transcript. And here you can see the location. These are the genomic coordinates where this gene exists. So this is the upstream sequence. So this is the genomic coordinate for this position here. And then this is the genomic coordinate for the position here. This is, and as we mentioned before, this is actually a gene on the reverse strand. So what we can see at this end over on the right-hand side of our screen, this is the most 5' exon where the start codon exists. So what we could do is we could just perhaps alter this these coordinates so that we are viewing an extra 2,000 base pairs upstream. So you can see here, now we have an extra 2,000 base pairs that we have here, 2 KB perhaps. And then if we want to retrieve that extra sequence, we can click on the export data button over on the left-hand side. And again, you can change the coordinates slightly so that you are retrieving the exact sequence that you want to. And then you would just click next to download. So then you can download in text, compressed text format, or you can even view the sequence in HTML here. You can see it in the web interface. There's one comment, yeah, I think I might have misspoke. I think I might have said transcription. But yeah, so yeah, you're exactly right. So the translation start site is the ATG. So in the example that we were using, there were no untranslated regions upstream of the ATG. So you're exactly right. So two very similar questions that have come in here. One asking how can we show enhancer marks, and then the other one showing whether any information for transcription factor binding. So that's actually not any data that we make available through the Rapid Release Genome browser. As I said, it's a lightweight genome browser that we just have available, where we just have the some main data sets available. So the gene annotation, so the genes themselves, and then the repeat features, as well as the protein features for the protein coding transcripts, for example. So at the moment, there are no tracks or data sets around transcription factor binding in the Rapid Release Genome browser. This data is available through, if you're interested in other species, such as human or mouse, you can get this data through the main Ensembl website, www.ensembl.org. Obviously, that was not the focus of this webinar. So we've been looking at the Rapid Release Genome browser, but those data sets you can find for human and mouse through the Ensembl Genome browser, ensembl.org. Okay, thank you very much. So if there are no further questions, then yeah, we're finished for the day. Thank you very much for attending. I hope you found it useful. If you do have any questions in the future, please don't hesitate to contact the Ensembl help desk, and we'll do our best to help you using the help desk.